To understand protein synthesis, we first need to understand DNA structure. And this is the basic structure of just a small fragment of DNA. It's built like a ladder in a double helix form. So all of this is one big strand of DNA, where this would just be like a little fragment, like that point right there. DNA is built up of 4 subbases: adenine, thymine, cytosine, and guanine. And where we get DNA from is deoxyribonucleic acid, so DNA right there. Now that we understand DNA structure, we can understand DNA replication. And to replicate DNA, you would take your sugar phosphate backbone, that's what this is right here, sugar phosphate backbone on both sides, and you would take one side of your DNA, line it up, all the subbase pairs with the sugar phosphate backbone, and then you would take the other side of your DNA, line it up, and it would correspond with its matching subbase pair to make 2 strands of DNA that are identical. But when we actually want to make a protein, we need to send this information, this blueprint, and that's what DNA is, it's a blueprint. We need to send that outside the nucleus into the ribosome to line up amino acids to actually make a protein. And that step is called transcription. And in transcription, we would take half of our DNA strand, so let's take this half right here, and we would make a sugar phosphate backbone with T, A, C, T, T, A, C, G, C. Now, RNA is similar to DNA, except with the exception that there is no thymine in RNA. So instead of thymine, we have what's called uracil. So uracil replaces thymine. Uracil is obviously shown with a U. So when we're matching up our DNA, I'll do it in a different color. When we're making our RNA strand, we would match the same, but now adenine, instead of matching with thymine, it would match with uracil. So thymine would still match with adenine, because RNA still has adenine, but now adenine would match with uracil, cytosine would still match with guanine, and then so on and so forth. And that is transcription. That's creating the actual strand of mRNA. But now when we want to send it outside the nucleus, now we can send this outside the nucleus now that we've created that. And we can start with a process called translation. And during translation, we send this mRNA strand, which is ribonucleic acid, RNA, or mRNA, messenger, for messaging to the ribosome. We send this, and I'll write it horizontally just so it's easier. A-U-G, A-A-U, G-C-G. Now that we have our mRNA strand running through the ribosome, we can match it with the appropriate amino acids that have the corresponding code for it. And the reason I did it in 3 pairs like that is because this is read, mRNA is read in 3s, which is known as codons. Codon, codon, this would also be a codon. The most popular start codon is AUG. Now this would have millions of subbases before it and millions of subbases after it, but for teaching purposes we'll just deal with this little section of DNA, or RNA at this point. So now that we have this running through the thing, and it finds its first AUG, which is the start codon, so we can start making the protein now. So we have, now we're in the ribosome. We have amino acids floating around the ribosome. This would be amino acid 1. And amino acids have what's a different form of RNA, which is called tRNA. And that would obviously have the corresponding subbase to the codon on the mRNA to line the amino acid up in the proper position to actually make that protein. So it knows, it pretty much gives the blueprints which amino acids to line up to make that specific protein, whatever you need it for. So this would obviously have A would pair with what? If
U.
A would pair with U, right? U would pair with
A.
And G would pair with
C.
C, right? Exactly. So that would have a code UAC on it. And then this one, amino acid 2, A would pair with U, A would pair with U, and U would pair with A, right?
Mm-hmm.
And these would come down and line up all in a row above the mRNA until you had a bunch of amino acids lined up to actually make a protein. And once you've filled that, and then there's also a stop codon, which we won't get into, but once you have enough amino acids lined up, then you can have that protein, you can send it out of the ribosome, and then you can do whatever you intended it to do with it, whether it be like muscle fiber or something like that, right? So now that we understand that, that's what's supposed to happen in a normal strand of mRNA and a normal protein synthesis, right? But sometimes that doesn't happen correctly, and you get what's called a mutation. So let's say during the process of transcription, there was a mistake in the mRNA. So we have our same A, U, G, A, but like let's say instead of AAU this time, something happened and guanine got replaced with adenine. So it would be GUA instead of AAU, and then it would be GCG. Now the same amino acid, your start codon, you still have the AGG start codon with your tRNA on it, with the corresponding code UAC. That would line up properly right there. But now, instead of our proper AAU blueprint, which would line up with the amino acid UUA, now you would have an improper amino acid lineup, because the amino acid with the codon CUA would line up next to the proper amino acid. So this amino acid right here would have actually a mistake on it, and this protein wouldn't be folded right, and it wouldn't be a proper protein for that muscle fiber that we're looking to make, and that's a mutation.
Okay.
And that's how mutations happen. So if you follow me over here, so like let's say this is our DNA strand, and after transcription, we take our DNA helicase and we split this in half, and then we want to send this strand of mRNA from our nucleus through the cytoplasm into the ribosome to match with the amino acids. So we have here a walnut, AUG. We have a peanut, UUA, and we have a honeycomb, CGC. So this gets sent out of the nucleus, through a vesicle in the ribosome, into the ribosome, and then amino acids would come in with the corresponding codon on it. So this would be UAC, right? Would match up with that, with the corresponding codon. This would come in, the cashew AAU codon would come in, and then to match up with this codon. These are all codons, by the way. The amino acid 3 would come in with the GCG codon and line up with the CGC codon. And that's what happens in normal protein synthesis. But let's say during our transcription phase, something went wrong, and this peanut codon, UUA, got replaced with an animal cracker codon, GAU. When you come into through the same process, through the cytoplasm, into the ribosome, amino acid 1 would look for its start codon, AUG, which is the walnut. That would line up fine, and the proper protein that we need to make would have amino acid 2 normally coming in and matching with the peanut codon. But now, because of the mutation of the animal cracker, this amino acid doesn't actually get placed there, right? It would look for a different amino acid that would match with GAU, so it would be CUA, and that amino acid would come in, and that would be the mutation. So yeah, that's protein synthesis. Yeah, that's about it. Hope you enjoyed my lesson.